So, session 2. Obviously, we've heard a lot in our earlier talks about the value of genomics and also in terms of how that is an innovation in our sheep breeding programs to increase genetic gain. To do that, of course, you need a reference population, so that will be what our session will be about today. Looking at, I guess, not only the existing traits that we're currently measuring as sheep producers, but also as industry, what are we doing in investing in new traits, and also different types of information that we collect through industry partners. George, can you come and take a seat, please? That means like now. So, ladies and gentlemen, our first presentation after the lunch break is from Hamish Chandler. Hamish is the Genetics Program Manager for Meat and Livestock Australia, a role he's held since 2016. Prior to that, Hamish started with sheep genetics in 2009 as the Land Plan Development Officer and managed the sheep genetics team from 2013 through to 2016. In his spare time, Hamish and his wife Leanne run a series of livestock businesses around the New England, and Hamish is here today to talk to us on a presentation titled The Resource Flock, an industry reference creating data. Thank you, Hamish.
Thanks, Luke. I was particularly proud of that title. It was quite informative, wasn't it? So I was going to start out with some fairly boring strategy stuff around, why are we investing in this space, before just giving you an update where we're at with resource flock projects. What are some of the things that we're now trying to develop and get in place to fill in some of the gaps that are in our reference populations, and what are some of the things that we're going to be thinking about investing in and getting in place over coming years. So I guess to start off with, we have a new red meat industry strategic plan in place. Red Meat 2030 was launched last year. I guess that took in the order of 10 months to develop. There was something like 300 stakeholders involved, producers, consumers, representative organisations, a whole range of organisations had an input into Red Meat 2030. But really, our vision is around doubling the value of the Australian red meat sales and being a trusted source of high quality protein. When that comes back to what's MLA's strategic plan, so MLA's strategic plan through to 2025 obviously draws heavily on Red Meat 2030. And it's about what are the things that we're working towards to align with that strategic plan. What are the things that we will be working on as priorities in our strategic process? And then following on from that, we then break it down into an annual investment plan. So people like me have got KPIs to work towards and make sure we achieve every year and continue making progress towards the broader industry objectives. So just to pull out, I guess, again, what the objectives are out of Red Meat 2030, out of our 2025 strategic plans. As I said, our objective really is to double the value of Australian red meat sales, make sure that we are the trusted source of high quality protein. There's a focus there on quality product quality, product attributes, including animal health, welfare, and environmental credentials. And our purpose is really to foster the long-term prosperity of the Australian red meat and livestock industries. From a genetics program perspective, I guess we launched the National Livestock Genetics Consortium back in 2016, and our target there, really in line with what the current strategic goals are, our target that we set back then was doubling the rate of genetic progress in the commercial livestock industry. And to do that, we've invested in a number of areas. We've had several project calls over and above our core research, developing analytical software and that sort of thing. And we've had several project calls and over the last few years have supported an additional 45, 50 genetics research projects. The bulk of our investment across beef and sheep really comes back to reference populations and how do we make sure we've got the data sets there to inform genomic predictions going forward in all of our different genetic evaluations. So our investment in reference populations really is the lion's share of what we're doing in the livestock genetics program of work. The next biggest component is around adoption and trying to make sure industry knows how and why to use those tools so we can continue driving progress in industry. Across MLA, you might be aware that there's a number of different call processes, prioritisation processes to look at what sort of research does industry think we should be supporting going forward. Coming out of that, there's been a number of areas of work that have consistently come up as high priorities but haven't necessarily been particularly well supported by some of those call processes when it comes to what sort of projects are being brought forward. So there's been a number of strategic partnerships developed over the last year or two to try and address those key issues. The ones that are relevant to this group today really are around the sheep reproductive strategic partnership and the carbon neutral pathways, in addition to the work that we're doing through the National Livestock Genetics Consortium. So you may have heard of MLA's CN30, Carbon Neutral by 2030 strategy, and as part of that, what are the different components or pillars that you can work on to improve industry's likelihood of achieving those aspirational targets. And I guess from a livestock genetics perspective, the piece that I'm interested in coming out of all of that is the greenhouse gas emissions avoidance part of it and how can we contribute to that as part of the genetics program. So recently, there's been, as part of that emissions avoidance partnership call, looking for projects, looking for collaboration in that space. A whole bunch of words up there, but the bit that I really just wanted to bring to your attention, I'll come back to in a little while, is the box in the middle there around low methane livestock. So when we start talking about reference populations, things like the resource flock, how do we start looking at some of the other priorities that have been raised by industry, some of the other work that's being done elsewhere in MLA. So I'll come back to that slide in a little while, but there's been a project call, there's been several projects coming in. The one relevant to this group again is a project there about selecting more methane efficient sheep. On the sheep reproduction strategic partnership side, so time and time again through the various regional advisory councils and the consultation process, there's been issues raised about how do we address lamb production through increasing weaning rates, decreasing mortality. So in that partnership that was formed, it was really looking at addressing 4 key pillars to try and improve those things. The top 3 boxes there are really around improving adoption of existing technologies, so best practice management, human social factors that affect adoption, enabling technologies, but underneath that, basic R&D. So again, there is scope for us to align much better with other call processes, other research that people are wanting to do to make sure that there's a really good link between what we are doing in the genetic space and how industry should go about increasing weaning rates, decreasing mortality. You've already heard from Andrew Swan, work that's been done within AGBU and the sheep genetics system to work on exactly that. So they have had a call process. I guess, unfortunately, from my perspective, there hasn't been any genetics-specific projects supported out of that process just yet, but we need to keep in mind that the work we are doing actually helps address a bunch of priorities across industry. From the National Livestock Genetics Consortium side of things, the future R&D priorities that are still being brought forward by the task force in that space, again, sustainability and welfare traits, particularly things around methane emission traits, heat stress, body condition score are some of the key things that members of the task force are raising as priorities that we should be doing more work on. Improved phenotype collection, so particularly around things like date of birth, reproduction, trait collection. You know, what sort of technologies can we use to make people's lives easier to record required data that we can then feed into genetic evaluations to get a bigger proportion of the national herd and the national flock recorded and into genetic evaluations for those sort of traits. And linking genetics through to consumers or consumer outcomes continues to be a high priority. We have done quite a bit of work in that space over recent years, but are continuing to look at how else do we further support that. Outside of that, there's also some priorities around genetics data platforms. We're doing probably more work in that space for sheep than anywhere else, and Peter's already been speaking about that this morning. And the other one was multi-breed genetic evaluations. I think that's probably more the beef industry trying to catch up to where we are in the sheep industry. So to bring that back to what does that mean for reference populations, again, I probably don't need to remind you guys about the basic idea of genomic predictions. You've probably heard it many times, but essentially we need to have all of the measurements, phenotypes for traits that we're interested in, plus genomic information on those animals. Look at the associations between what you see on the DNA, on the genomic test, what you see in those measurements that, from that reference population, and then that gives us the ability to go and genotype animals out in industry, say, we've seen that pattern on DNA tests before, it's associated with this sort of performance. Some of these slides definitely are not new. I actually used exactly these slides in the previous Leading Breeder Forum 2 years ago, but just to bring that back into perspective so I can then go on to say, what have we done since that point in time? A reminder, I suppose, what's the key to genomic success? To make sure it actually has an impact on driving change in industry. You know, the uptake and impact of genomics really comes back to how high can we get the accuracy of those predictions and the cost of doing that work. So to me, you know, anything that we do the majority of what we do in the livestock genetics program is really related back to accuracy of breeding values, and the better we can get that accuracy, the more frequently people are making the right selection decision and the faster we can make genetic gain. So when it comes to genomic selection, how high can we get that accuracy of prediction has a direct impact on the rate of genetic progress. To do that, we need large reference populations, up around 5,000 animals with records for the trait that we're interested in. We need to continue maintaining and refreshing those populations over time. So, you know, an additional 400 or 500 animals per year to make sure that it's the genomic predictions are really reflecting the genes that are out there in the current population. And a genetic evaluation that combines both genomic information and phenotypic information, data that people are capturing on farm and elsewhere in industry. So most of you would be aware, but we started on the resource flock sort of approach back in 2005 with some sheep genomics work. That went on to the sheep CRC, information nucleus flock, then morphed into MLA's resource flock starting in about 2012. So we've got a significant sized reference population for a lot of traits that are fairly routine we've been measuring over that period of time. With the newest project that Julius is still managing for us, we're still continuing to record in the order of a couple of thousand lambs a year from birth through to slaughter. So continuing to grow and refresh our reference population for the key traits that we already have genomic predictions based on. Last time we spoke about this stuff, we did go into, so what are the options for how to capture reference population information? Reference population data to support analyses. So our options are structured research projects like the resource flock, where we know we're going to get good quality data We can build into that measurement of hard to measure or novel traits much more easily. We get to plan matings to reflect the diversity across industry, but they do tend to be expensive on an animal basis and reliant on ongoing project funding. Breeders' flocks, your flocks, are the other place that we can capture reference population data and have that contribute to the analyses. Really good for capturing big volumes of information that people are collecting for routinely measured traits anyway. So people are often doing that already. All we need to add to that is a genotype. Data is obviously usually of reasonable quality. In some cases, like repro traits, it's easier to measure that in your businesses than it is in a research project. But there are some downsides, particularly for hard to measure and novel traits. Things like eating quality are very difficult, obviously, to measure in a stud breeding context. We need to have an understanding of data quality coming out of those individual flocks. Gus has already mentioned and thrown to Sarita about some questions about data quality indexes, and that's one of the reasons behind that work being done. So the other option, I guess, is commercial industry data coming out of abattoirs, that sort of thing. So much cheaper on a per animal basis. We can access massive volumes of data and across the supply chain, but data quality can be a limiting factor. What do we know about the fixed effects that we normally want to deal with? Date of birth, dam age, management groups, all that sort of thing. How well are these animals that we're using measurements on related to the rest of the population? Other biases that might be included in those data sets. So you can think about that in context of how many records do we need to inform a particular level of accuracy on our genomic breeding values. So if you think there about the accuracy prediction and the number of animals we need to achieve that, if you're wanting to achieve at least a 40% accuracy, the chart there shows you if we've got a relatively modest heritability of a trait around 30%, if we're collecting really good quality data that supports a decent heritability on our trait, we probably need in the order of about 3,000 animals with records. If using commercial animal data, for example, there's more biases or there's other fixed effects that we can't account for, that means that the trait we're measuring actually has a lower heritability. You might need in the order of 6,000 animals in your reference population to get the same accuracy of prediction. So from an investment perspective, what's our best approach to go collecting data that means that we're going to get a good return on investment for industry in terms of accuracy of selection and how much we're paying for that. So what's happened since the last Leading Breeder Forum, when I last went through all of that sort of stuff with you? Since that point in time, as I said, we've continued growing and collecting data in our resource flock project, with another couple of thousand animals being recorded a year for basic on-farm traits through to abattoir traits and lab-based measurements for eating quality. Since that time, we've obviously identified that one of our gaps is in what's actual consumer feedback like on pieces of meat. What's the sensory information tell us? So there's been work going on. Pete's here in the room with us, trying to do more to link genetics through to consumers, looking at sensory evaluations of lambs out of the resource flock. So over the last couple of years, it has been delayed a little bit with COVID and all the rest, and being able to get panels together for that work, but looking at having measured more than 3,000 odd animals for those traits. Jess Perovic will be speaking shortly about the MSA Mark II model for sheep. A lot of this data has been not just useful for us further developing genetic evaluations, but also for work around how do we do a better job on MSA for lamb at the same time. So that's one thing that's progressed significantly since we last discussed things at Leading Breeder. Reproduction traits. So as I mentioned, there are some traits that are much easier to measure outside of that resource flock structure. The resource flock is based on base ewes that we really don't know a lot of pedigree about. We then know, obviously, all the pedigree details for the sire side of things, but that doesn't really enable us to capture reproduction data within the resource flock. So over the last couple of years, we've been working with breeders, with stud breeders who have got a track record of being able to demonstrate good reproduction quality information, and we've been genotyping a bunch of animals, over 13,000 animals from 26 flocks for maternal breeds, up around 4,800 animals in the Merino database from 13 flocks. So that data is continuing to come into the database and will definitely help underpin our ability to do genomic predictions for reproduction traits. We will be doing more of that work, so definitely keep an eye out for those calls for expressions of interest as we go forward. And similarly, we've been working with breeders to capture lamb kill data and trying to evaluate some of those ways of how else do we capture reference population data outside of that normal resource flock structure that might give us more cost-effective ways of measuring information. So we have done work collecting data both for surplus seed stock animals and commercial or progeny test sort of lambs, and continuing to do more of that sort of work going on as well. For some of the other priorities that I was mentioning around sustainability, for example, we do now have our first project in place to start capturing methane and other gas trait emissions out of resource flock lambs. So that was when the shed at Katanning was first being built. It is having a whole bunch of methane emission measuring infrastructure put into the shed so that lambs that come out of the Katanning research site can go through and have methane emissions recorded on each of those lambs. So that is a one-year project at this point in time, but looking at how we support that over the next 4 cohorts as well to make sure that we're starting to capture sustainability traits on our resource flock lambs. As I mentioned before, through that emissions avoidance program calling for applications, there were several genetics-related projects that came in. One of those was a sheep project, so we are currently going through the process of seeing whether we can get support for that project. But again, based on not just resource flock lambs, but sheep out in breeders' flocks as well, how do we go about measuring feed intake and methane emissions on those animals in a way that we can get out of that really controlled environment and having to have a unit to stick over the top of each and every sheep. So can we develop mobile methane measurement facilities and do that a lot more broadly across industry, make sure that we're able to develop genomic predictions for methane ASBVs. To come back to the Nlgc approach, as I said, the Nlgc priorities, I think that link through to reference population work, really have been around things like methane emissions was one of the outstanding priorities, things like heat stress, body condition score. So they are things that, one, we've got some projects coming through that will start addressing that. Two, we will be planning on addressing some of those with future call processes where we go out asking for researchers to put out expressions of interest, and how do we go about looking at what sort of new traits should we be investing in that space. Brad Hine will be talking shortly about work that he's been doing elsewhere in the immunocompetence field. I think that's something that's really interesting work that we should pay a little bit more attention to. How do we use some of those sort of concepts to breed animals that are more resilient and likely to hold up to more environmental challenges as we go forward. So I guess just to summarise where I've been, what I've been trying to get across over the last little while. So our reference populations for existing traits are continuing to develop. We're still adding another couple of thousand animals to those on an annual basis. We have started trying to address those different pathways to capture more reference population data through partnerships with stud breeders and commercial breeders, and we're continuing to work towards developing better links to link genetics through to consumer outcomes, consumer expectations, and we'll continue developing ways to address industry priorities for sustainability and welfare traits in particular. I'll pull up there and see if we've got a few minutes left for questions.
Thanks, Hamish. This is a bit where we clap, guys. Yep. Thanks.
Thanks, Luke. That was getting a little awkward.
Okay, so a few good questions to start, Hamish. First one around the how of measuring methane and what sort of feeding are they using?
Yeah, you're probably asking the wrong person. The right person's probably Karis, who's watching from home today in Western Australia. But at this stage, the initial work will be done using containment units in the sheds at Katanning, so you can have animals in there for a period of time to collect the gases that they are emitting. They will be basically on a pelleted ration to be able to have them in the sheds to collect that information, which is one of the reasons why people like Julius are looking at other opportunities to measure that out in the paddock. The beef industry is tending to use more of the green feed type units where they cattle have you put an attractant in them, cattle go up to those units out in the paddock. As they put their head in to feed, there is units over the top that draw the gases in and do an analysis of the gas out in the paddock. So I think that is somewhere where we would like to get to. At the moment, it is very much shed confinement feeding based. Those units are quite expensive, so how do we do that in a cost-effective way? How else can we develop that over time is a research question.
Okay, so following on the theme of trait measurement, Hamish, feed intake, is that currently being recorded?
So that is part of the work that will be done at the Katanning site at the moment. So that will be recorded for the first time with this cohort of lambs, doing both feed intake, as I said, on a pelleted ration, and doing methane recording on those same animals at the same time. Okay,
so this is a good question. I do like this. So obviously one of the models that we've seen presented today is around, on-farm data contributing in the way of reference populations. The value of that versus a very structured resource site.
Yeah, so it comes back to what's the cost of collecting that information as opposed to the accuracy that you can derive from it. So there's quite a bit of work that's been done. We've got other projects that Queensland Uni are working on in the beef space, for example, where capturing genotypes and phenotypic observations on 50,000 commercial beef cows, for example. And on a big data set like that, there are limitations because of not being able to account for fixed effects, but with enough data you can overcome that and still derive information with useful heritabilities and derive accurate breeding values as a result of that. So to me it sort of comes back to there's a number of different ways that you can capture that data and use it to predict breeding values, which allows us to account for the most biases, but then at the end of the day it comes back to a cost effectiveness sort of approach. So do you need to capture 10, 20,000 records of lower quality data and genotype all of those animals, or is it more cost effective to capture 5, 10,000, 2,000, 5,000 records where we've got a lot more information, a lot more fixed effects, so ultimately what do you get the highest accuracy at the lowest cost from? So I think over time it will definitely be a combination of those approaches. So I think the R&D that we're going to have to be starting to look at is how do we actually integrate some of those different approaches into the one genetic evaluation to get the most cost-effective outcome.
Great answer. Okay, so just with your program manager hat on here, how are we measuring that the rate of genetic gain in terms of doubling, and how likely are we to hit it by 2022?
So that's the $1 million question. Probably more than $1 million. But at the end of the day, we will still be using rate of genetic progress out of our genetic evaluations to underpin that, but trying to look at how well is that being adopted across the commercial part of the industry. So it's a combination of the rate of genetic progress that we can demonstrate out of the major genetic evaluations and the adoption of those technologies. We did the Ipsos survey back in 2016. That was a survey looking at 2,000 producers around the country, half beef, half sheep. At that stage, the beef producers were telling us that about 18% of beef producers were using EBVs as a basis for their bull selection. I think sheep producers were somewhere between 14 and 16%. The more recent surveys that we've done off the back of some of the adoption-based projects that Clara has been leading, the most recent feedback, it was a much smaller survey, 600 odd producers, but that indicated that we're more in the order of 48% of beef producers were using breeding values and 44% of sheep producers were using breeding values. So we can demonstrate that we have been making ongoing improvements to the rate of genetic change. We're still driving that quite effectively, but we're also getting to the stage where we can demonstrate that there's a much, much bigger proportion of industry utilising that information to drive progress within their businesses as well. So I think, yes, if you put those two things together, we'll get a pretty decent understanding of the impact we've had on industry. And I think particularly with that growing use of information out in industry, we should meet those targets.
Great answer. All right, I'm going to pull things up there. Hamish, congratulations, right on time.
First time for everything.
There is a first time, so let's celebrate that, guys. Give it up again.